kinetics. And so, this is just basic things about reactions. And so remember that, over the course of a reaction, what's going to happen is the substrate level will go down and the product amount will go up, right? And that's what this graph shows, right? We have substrate going down and product going up. And if we're looking at that reaction, right, and this is catalyzed by an enzyme, if we add more enzyme, right, we're not adding more product or anything, we just add a bunch more enzyme, what's going to happen is that substrate will get converted to product faster because there's more enzyme there to grab onto the substrate and convert it. And so we can look at the rate of that reaction, right? So how fast this is progressing. We call this velocity, right? So how the velocity would increase as we increase enzyme concentration. And so as we progress through a reaction, if we add more substrate, right? So now not changing enzyme amounts, but changing substrates amounts. What we can do is we'll see that velocity will increase with more substrate to a point, right? So initially, velocity will increase rapidly as we increase the amount of substrate, right? Because there's more and more substrate, so it can actually do the reaction until we get to a point, right, to where we're at the absolute maximum velocity, and that's what we call Vmax, right? And that's where we're completely our enzyme is now saturated with substrate. And so that'll give us this hyperbolic curve, right? Where initially velocity is increasing very rapidly, but then as we keep increasing substrate concentration, that'll slow down till we reach maximum velocity. And so when we actually are going to look at enzyme kinetics and we try to study them, it's difficult to study them at those points where we're increasing substrate concentration. So if we're going to look at, just the overall how the enzyme's working, we work under steady state conditions. And so this is the point where the substrate is going down, the product is coming up, but the actual amount of enzymes and enzyme substrate are not changing. And so those levels will be constant. And so that's this time here is where we would have steady state conditions. And so one thing we had an equation on for our quiz, and you'll have another equation on it for the exam, is in the Michaelis and Mitten equation. So you should be able to use this equation to calculate, any of these terms if I were to give them to you, right? Calculate initial velocity, calculate Vmax or Km or the substrate concentration. But it's not going to be any more complicated than what was on the quiz, right? This is a long time ago. So if you can understand how to do that, I think you have what you'll need for the Michaelis-Mitten equation on the exam. And so, right, definitely understand though what Km is, right, and where it's the Michaelis constant, but it's where it's this concentration of substrate where we're at half max velocity. So it's right here, because this is going to help us understand, right, things about inhibition, and when we look at the next different curves. All right, and so I went over a little bit in class about how we actually do enzyme kinetics and how, what we're going to do is a series of reactions over time at a certain substrate concentration. And then we're going to do another series series of reactions over time with another substrate, higher substrate concentration, and then we keep doing that, right? And then we can plot those values. We could plot them and get the Michaelis curve, but it's difficult to actually then use that to determine any of these numbers. Nowadays we use computers and we can do that, but when this was done before, it's really difficult to do by hand. And so we use a double reciprocal plot, these Lineweaver-Burke graphs. And so when we do that, now this can actually, we can just use the graph and look at it and be able to calculate the Vmax and Km. And that's because where our line crosses the y-axis, this is 1 over Vmax. And if we extend our line into sort of hypothetical values, right? Couldn't really have negative values. The point where the line crosses the x-axis, this is -1 over Km. And so we can just look at one of these curves and we could get these values. And so I want you to be able to do this just before, same as before as a quiz. On the quiz, is that if you were given, one of these, a plot like this, could you tell me what the Vmax of the Km was? Yeah. So are there questions about what you need to know for actual calculations or doing the short answers for enzyme kinetics?
So do we actually need to know the inverse of Michaelis-Menten equation, or is it sufficient for us to know the intercepts?
No, the intercepts.
Okay, thank you.
Yeah. Okay, so then we talked about how we or how enzymes can be inhibited, right? And there are different types of enzyme inhibitors, and the two, just broad types of inhibitors can be reversible ones, where it's going to be non-covalent interactions, the inhibitor will be able to come off, and irreversible ones. So now we're going to have things with covalent bonds that once the enzyme's inhibited, it's inhibited. And we talked about how really good reversible, technically the reversible inhibitors are the transition state analogs, right? Because they remember how enzymes are working and stabilizing the transition state. And so if you have an analog to that transition state, it's going to bind very well into the active site. And we talked about, several types of inhibition and mechanisms of inhibition. And so these you're definitely going to know for the exam, not just, for multiple choice and that, but also I said, I was pretty specific about it. You're going to have to take a curve and a double reciprocal plot and be able to draw lines for each of the different types of enzyme inhibition and what we would see for the reaction. And so the one major type of reversible inhibitors are competitive inhibitors, and these bind to the active site. And we call these competitive because they're competing for the active site with the substrate. And so one thing about them is that if you add more substrate, you can outcompete the inhibitor. But if we look at one of the Michaelis graphs, you can see with the inhibitor, what do we do? Right, this starts to come down. And so what we see is an increase in our Km. We don't see an increase in Vmax. It's not changing how the enzyme functions and it's able to work. It's just competing with our substrate. And so we see an increase in the Km, but no change in Vmax, right? And often we call this what we see with an inhibitor, we call it a parent KM because it's what it appears to be. It's not, if we get rid of the inhibitor, the KM is back to normal. But if we look at a double reciprocal plot, right, we can see a change in the slope of the line when the competitors and the competitive inhibitors there, but we don't see a change in where this line crosses the y-axis, right? Because we're not changing Vmax, we're only changing Km. You will be asked something about calculating either the alpha or the Ki for the competitive inhibitor. So I would take a look at that. It won't be anything super complicated, but you should understand that Ki is an inhibition constant, right? And so the lower the value of the Ki, the tighter the inhibitor binds. My slides are not changing. Okay, so we covered that transition state analogs are really good competitive inhibitors. And so knowing why, it'll probably be asked as a true-false question or a multiple choice. Okay, so another type of inhibition is uncompetitive inhibition. And so these types of inhibitors only bind to the enzyme substrate complex. So we do have substrate binding. It's not competing for that site. We actually have to have the substrate there. And in this case, when we look at our double reciprocal plot, right, what do we see? The lines are parallel. And so we're not actually changing slope, but we are changing, right, Vmax and Km and the apparent Km. And then for non-competitive inhibitors, these are binding at someplace other than the active site. So the other side of the enzyme or in a different place. And depending on the non-competitive inhibitor, sometimes the substrate has to be there and already bound. Sometimes it can't be. It's just about that inhibitor and what structure it's going to bind to in the enzyme. And in this case, we see a change in the slope, but what we see is that Km hasn't changed, right? So in this case, the substrate is still able to bind the active site and it binds it. But what we're going to do is change the activity of the enzyme. And so in this case, adding more substrate doesn't let the enzyme overcome the inhibitor.
Dr. Reynolds,
So, yeah.
Sorry, do you, if you're done, do you mind going back to the transition state slide?
Yeah.
Once you're done, so.
Okay, so the only thing, I'll just so you know, and I think I said this in class, so this can be called mixed inhibition as well, but we're going to call it non-competitive inhibition. Okay, what would you like to talk about for transition state analogs?
So we know that they make better competitive inhibitors than substrate analogs because they're better at blocking the active site.
So they make transition state analogs make better inhibitors than substrate analogs because remember what the enzymes are doing, they're stabilizing that transition state. And often they hold on to the transition state. It's usually the state where in the reaction, it's the point where it's going to hold on the tightest actually. And so if it's in a transition state analog, when it binds to the active site, it binds there, but then often we use things that they're analogs that the reaction can't proceed. And so we're having it so it binds, with the highest amount of strength in that reaction and then still can't go forward. So it's not going to really want to let go because of just, the ability to release it.
Okay, thank you.
You're welcome. Does that help?
Yes, it does. Basically, you're saying that the transition state, you're allowing for strong binding, which is but strong binding with a ligand that isn't able to proceed with the reaction.
Yes.
Okay.
Okay, so that is where I think let me check for the short answers, we were ended with enzyme kinetics. So I totally realize that this was a long time ago. And so that's why I'm kind of being specific about what I want you to know for the short answers. Okay, and then we switched gears quite a lot to start talking about DNA, right? And so this goes to chapter 3. And so we talked about the nucleotides and the structure of the nucleotides. But first we talked about the nitrogenous bases that they contain, and DNA containing adenine, guanine, cytosine, and thymine, and RNA containing adenine, guanine, cytosine, and uracil. And so I'm not going to have you draw these. I would suggest you know the numbering on them, and where they attach, where that base attaches to the sugar, like which position. And so we have this, in addition to the base, we have a pentose sugar, right? And so the difference between DNA over here and the sugar in RNA is that one hydroxyl group, right? And so we have in RNA, right, which is ribonucleic acid, we have OH is at both 2' and 3'. And then in DNA, it's a 2' deoxyribose. So deoxy, so we don't have that oxygen at the 2' position. And remember, we call this prime, we number these 1', 2', 3', just to differentiate them from the numbering of the base, where we don't use the prime. And it's that way if you're looking at the 1 position versus the 1', it's just so you can distinguish them. We did talk about the pucker a little bit. I don't worry about this for the exam. I did that in class just to kind of let you know that it's not a perfect ring. It's not perfectly flat, right? We have a pucker. But you should know the bond between the base and the sugar, and that right, it's a glycosidic bond, and in this case it's a beta N glycosidic bond, right? So it's beta linkage, but we have it to a nitrogen, and so definitely understand that it's a glycosidic bond. Okay, and then right, DNA, it's not single-stranded, it's in double-stranded helix form. And what we talked about, and I didn't say it explicitly at the beginning, but I got to it later, is that this slide here is covering the B form of DNA, right? So here are your major features of the B form of DNA, right? So to get a double helix, you have 2 strands that hydrogen bond with each other. They run anti-parallel, right? So one strand's 5' to 3', but then you have to flip the other strand over. And so going from the same end, right, it's 3' to 5. It is a right-handed helix. I didn't show it here, but if you go back to we talked about helices and proteins, I showed how to tell a right and a left-handed helix. The diameter is about 20 angstroms. You should definitely know that there's 10 bases per turn. And then we get that base every 3.4 angstroms. When we get the twisting happening, we form these two helix and we get these two grooves. So we get this major groove and this minor groove. And so what this means is if we just look at the distance between the strands, right? So here we have a much wider distance versus here we have a shorter distance. So this is the minor groove and this is the major groove. And when they bind, right, for hydrogen bonding with between our bases of the two strands, that means our sugar and phosphate are on the outside, right? So we get this backbone, the sugar phosphate backbone here. And this is going to give, right, the outside of the DNA a negative charge because we have that phosphate. And then on the interior, right, we get those bases stacking on top of each other. And I think we can see this here, right? And so this is perpendicular to that helical axis, but this stacking interaction is really important, right? So these bases are all stacked. And then you can see it here well too. They're all stacked in the middle, right? So it's base stacking in the middle, sugar phosphate, backbone on the outside. And so how is the double helix stabilized? So we do have hydrogen bonding. It does contribute to stabilizing the helix, but that base stacking and interactions with water, so really you call this a hydrophobic effect, are extremely important, probably more important than the hydrogen bonding for stabilizing the helix. Right? If you just look, van der Waals are always, the weakest of the forces, but when you have these base stacking, is you get a large number of bases, right? Remember we talked about that with protein. You get a very large number of van der Waals interactions, then it becomes significant, right? And especially if you look down, right, look down the end of the helix, you can see this is not empty, right? These bases are stacked on top of each other, filling up that space. And then also we talked about the hydrophobic effect. And so water molecules are tightly associated with the helix, and both in the major groove and in the minor groove. The really difference is in the major groove, where we have more space, and also with those interactions, we get lots of water. It's more disoriented or disordered. And then in the minor groove, we get a really rigid, like single file of water molecules in that minor groove. But this is stabilizing that helix. And in fact, when you start to take DNA and you start to add it into different solutions and with different amounts of polar molecules, you can see that as we have less and less water, these things actually start to come apart. And so the interaction with the water molecules, more and more, we find to be more and more significant for stabilizing the helix. And so I'll probably ask you a similar question to like on the quiz about knowing these forces and what are the most significant of the forces holding the helix together. Okay, and then that brought us to DNA replication, and DNA replication occurring in a semi-conservative way. And it occurs this way because each of the strands in the helix serves as a template for a new strand. And so if we have two strands of DNA and we separate those, what we can see is that using our base pairing rules, we can just take this template strand and know exactly what to put right on the other side for the new strand. So if it's a C, you add a G, and A, you add a T. And then after you finish this whole strand, what you have is one old strand and one new strand, right? And then you'd have that for the other one too. You'd have one old strand and one new strand. And so that's why it's called semi-conservative. So out of these two we get, each one has half that's been conserved. And so there were other ideas before about conservative, where somehow these separate here and somehow we get two new double helices, but one's all old and one's all new. There was another one called dispersive, where somehow some of the new double helices, some of it was old and some of it was new, and it was a combination. But really the mechanism shows us that it should be semi-conservative, and that's what we see. And so when DNA gets replicated, right, it has to first open up. And so when that opens up, it makes this like replication bubble, right? So we get these two strands separated, but it happens at one point, right? These origins of replication. And so that's where we initially start to synthesize that new DNA strand for both of those strands. And we call each of these points, right where the strands separated, right? This is the replication fork, because it's the split. And so we have two replication forks. The forks themselves move in opposite directions. Okay, and the enzyme that replicates the DNA, right, is DNA polymerase. And DNA polymerase always goes in the 5' to 3' direction. So what this means is it's going to take an incoming base, take the 5' phosphate and hook it up to the 3' hydroxyl of the base that's already at the end of the chain, right? So 5' is hooked onto 3'. So that's why we call it 5' to 3'. And DNA polymerase, in order to extend it, in order to put a nucleotide in, has to already have a pre-existing OH at the 3' position. That has to be there for it to do its reaction. And so when we go through replication, we have to have some kind of primer. We have to have something there for it to attach to. Remember where the energy comes from for this reaction, right? The incoming nucleotide, right? It's a triphosphate. And so the energy for this reaction is brought with that nucleotide, because we're gonna, let's just say, essentially hydrolyze this bond, right? So we're gonna release pyrophosphate. So it's like hydrolyzing ATP. We now can use that energy to create a new bond. And so as I always say, the energy is brought with the DNTP. And then so this bond that gets formed is a phosphodiester bond. So there's two ester bonds, and they're both phosphodiester bonds. And so all of the DNA polymerases we talked about all have a common structure. We talked about it kind of look like, or being said, it looks like a hand. And so the different polymerases do different jobs. And so in E. coli, they have about, at least we know of, 5 DNA polymerases, and then eukaryotes have, right now we know about 15. So you should know which polymerases, really I want you to know which of the replicative polymerases, and which one is primase would be good too for, especially for multiple choice questions. And so in E. coli, DNA polymerase 3 is the replicative polymerase. And in eukaryotes, they subdivide these tasks. So the leading strand, there's a polymerase for it, epsilon, and for the lagging strand, DNA polymerase delta is used. And we talked about the mechanism of DNA polymerase to make that bond, and that we had these 2 coordinating magnesium ions. And one of them, its job was to facilitate the attack from the 3' hydroxyl onto that alpha phosphate. And then the other one's helping to stabilize these other 2 phosphates for displacement after we get this bond formed here. And so these two will get let go. And so those magnesiums, along with the aspartates that are coordinating them, right? This is that transition state here. So together they're stabilizing this transition state for this nucleophilic attack. And so DNA polymerase, right, when it's going along, all enzymes, right, are going to do their things and sometimes release, and it's going to bind its substrate and then let go. But what we want for DNA polymerase is we want something to continuously replicate. It's not going to put in a base or put in a bring in a new DNTP, put in that nucleotide, and then fall off. And so an enzyme that can go lots of catalytic rounds before letting go, we call this processivity. And so DNA polymerase has what we call high processivity. So just even alone by itself, it does this okay, right? Compared to lots of other things, how we think enzymes grab their substrate, let go, right? Let go of everything. DNA polymerase in E. coli can do about 10 to 15 cycles before it lets go of the DNA. So this is already useful. However, there's something called the sliding clamp. And the sliding clamp is a ring that goes around, it's a protein ring that goes around the DNA and holds the polymerase on. So that as the polymerase is going on, holding on, doing 10, trying to let go, well, it can't because it's stuck there. And it's stuck there because it's literally hooked to it's going to bond to the sliding clamp, and the sliding clamp is stuck around the DNA. And so we have something called a clamp loader. And this is an enzyme that's going to use the energy from ATP to open up the clamp and hook it around the DNA and then let go of it. And so it used that energy to put on the clamp. Now DNA polymerase associates and doesn't come off. So it can do thousands of nucleotides before it comes off. Even still, with this, there are some polymerases like DNA polymerase epsilon that actually kind of has a little bit of the structure of the clamp. It has a little bit of a loop that can grab onto the DNA. So even without clamp loader, it's significantly better than most DNA polymerases. So does this make sense how DNA polymerase is going to keep its high level of processivity? We're going to have this additional protein, the sliding clamp, that it's going to associate with, and it's going to hold it on to the double-stranded DNA. Are we okay with that?
I'm okay with that.
Okay, thanks. Okay, so DNA replication happens, and I'm not covering a lot of it here, but we talked about leading and lagging strand and how, the differences between them. But as we're replicating, right, what's going to happen is the leading strand is going to just be able to go, and it's going to be able to get to the end, right? Because it already had one primer, and it can go all the way until it gets to the end of the DNA molecule and release. And then it's done its job. It's completed that whole strand. But on the lagging strand, right, we had to have lots of little primers added. And then we make it in short fragments called Okazaki fragments. And then those primers get removed, right, and then replaced with DNA. So what happens at both ends of a linear DNA molecule, right, as we're replicating, at the ends of the DNA molecules are replicating, what's going to happen is one of the strands can finish, but in the other one, the lagging strand is going to have a primer there that's going to get taken off. And if that primer gets taken off, then what you're left with is a little gap. And it's not enough, right? Whatever the length of that primer is, it's not like now we use a shorter primer and a shorter primer and a shorter primer. because that, that's not going to work. We're going to end up at some point where we can't have any shorter of a primer. And so we would still have a little bit of a gap at the end of the lagging strand. And so to fix this issue, we use an enzyme called telomerase. And so the issue with why we want to fix this is that over several rounds of replication and not replicating a certain number of bases, right? Because the primer gets removed, and then in the next round, a small bit will get removed, is that eventually we'll make it into genes and we'll start to have, right, an effect on what genes we're able to express because our chromosomes were shortened so much, we'll be chewing back the genes encoded in the DNA. And so to get around this, we use telomerase. And so what telomerase does is this brings along essentially its own template strand. But this template is made up out of RNA. And so the enzyme brings this non-coding RNA with it, and it uses a little short bit of it as a template. And then what it does is takes its own template and then puts in DNA on the other side and then keeps moving and adding in DNA. So it's extending this 3' end of the DNA, and it's doing that, right, with its own template. And so as it uses its own template and then moves along and keeps doing that, we get repetitive sequences because it's using that same template over and over and over. And so this gives our telomeres a repetitive sequence. And so as telomerase goes along, and that's what we see here, telomerase adds next, we get this extension, and this is flipped on the left compared to what's on the right. So it's just flipped over. But as we get that extension of DNA, well, now we have room to add a primer and start to continue replication. And because it's a bunch of repetitive sequences, it wouldn't actually matter that the next round maybe is a little bit shorter, because all you're doing is, say, missing one little bit of this repetitive sequence. And so this way if we were to do that, we would not be getting into genes. We would just be not replicating one of these repeats at the end of the chromosome. And so because telomerase is using its own RNA template and putting in DNA, this is called reverse transcriptase. So it's using an RNA template to make a new DNA strand. And so that's because what transcription is, and we'll get to it, but what transcription is using DNA as a template to make RNA. And so this is the opposite of that. This is using an RNA template to make DNA. And so we have this repetitive strand. This strand, right, is because if you look at the sequence our telomerase is using, this DNA has a high content of Gs. And then that would mean the other strand has a high C content, right? And so at the end of our chromosomes, we don't keep it, right? We don't keep it this way as this linear double-stranded DNA molecule. What happens is that G-rich strand gets itself, the end of the chromosome circles back, and that G-rich strand binds with the C-rich strand, and we get this circle. And then this wouldn't be that stable. And so we have a protein complex called shelterin that stabilizes, right, this circular structure. And this is called the T-loop, right, for telomere. So this T-loop gets stabilized by shelterin. So this is an extra credit question you had on the quiz. And so why we want to do this This is to protect us from some of these DNA repair mechanisms we have. And so one of those DNA repair mechanisms we talked about, right, was non-homologous end joining. And that's because if your chromosomes get broken, your cell wants to repair that. It wants to repair it fast, and it just wants them hooked together. It would like it to be perfect, but if you can't do that, just hook those ends of the broken chromosomes together. And we have this enzyme, or this enzyme that's going to recognize the ends of the free double-stranded DNA that would be broken. It's going to bind that, chew back some ends, and then some bases get added, and these get hooked together. And so now what we've done is grab the ends of this chromosome that were broken and hook them together. It's definitely not perfect because we don't really care. We just want to hook our chromosomes together because they were broken. And we talked in class about how, as you age, your chromosomes are older and they've broken over time. And so your cells are going to have like 2,000 or so scars from having this happen. So it happens a lot, and we need this repair mechanism. But if the ends of our chromosomes weren't in loops like this, that were protected, the one thing that would happen is that these proteins would recognize it, right, as free DNA, grab them, chew them back a bit, and hook them together. And so over time, what would happen is all of our chromosomes would just be joined together. And so we need this mechanism, but we also need our chromosomes to not all be hooked together. And so this T-loop structure, stabilized by shelterin, protects the ends of our chromosomes from having them hooked together by non-homologous end joining. Does that make sense? Maybe.
Yeah, that makes sense.
The other repair mechanism we talked about homologous recombination. And so in homologous recombination, after your DNA is replicated, you're going to have, right, a homologous chromosome right next to it physically. And so if you get a break, then you can use that chromosome that's not broken to repair your unbroken, or you can use the unbroken one to repair the broken one, right? And it has, right, its sequence is intact. And so when you do that, you get, sometimes we call this like flawless repair. And so you're going to get these without breaks, and you're not going to have those coup proteins that like chew back the ends and repair it, but they don't have a template. So they're adding things that you didn't have before. So in this case, we're using DNA we have to make this repair. So we have all the DNA that we had before, and we have the right sequence. Okay, so we talked about another reverse transcriptase. We talked about the HIV reverse transcriptase. And so our HIV has a single-stranded RNA genome. And so what it does, it's part of this group of viruses called retroviruses. And what they do is take their RNA genome and convert it to DNA. So HIV has an enzyme for this. So it's reverse transcriptase. And what it does is it uses actually a host tRNA, so a tRNA lysine, and it uses this actually as a primer. And so the host tRNA lyse is there, it works as a primer. The HIV transcriptase comes along, it takes RNA, and it makes a complementary strand, right, a new strand, but this one's of DNA. So we're doing RNA template for DNA synthesis. And it's synthesizing this new strand of DNA. As it's doing it, it actually chews up this RNA strand. And then it gets to the end, and now we have, right, single-stranded DNA. And it turns out that HIV reverse transcriptase can also use DNA as a template. So then it turns around and it uses the newly made DNA is a template to make, it uses the newly made DNA strand as a template to make the second strand of DNA. And so now we're to a DNA double helix from this one enzyme. And what then HIV does is it can take this and put it into your genome, right? A human genome, and essentially hide out as double-stranded DNA in the genome. And for a long time, people are interested in coming up with inhibitors for reverse transcriptase. And so we have had some for a long time. And initially they were nucleoside analogs. And so there were things that didn't have a 3' OH. So there are things that if this got put in by the enzyme, it would halt DNA synthesis, right? Because you couldn't extend the strand here or here on these two drugs. But people develop resistance really easy to that, or the HIV reverse transcriptase can develop resistance to that. And so people started looking for non-nucleoside analog drugs. And that brought us to this paper that we looked at in class. Right, so they did a screen and they found one of the drugs in their, or one of the chemicals in their screen worked as an inhibitor. And then this was not a nucleotide analog. So it was something else. They guessed it probably wasn't binding to the active site, but they needed to test that. And so they did these reactions we talked about, right? They did the kinetics. And as they added the inhibitor, right, what they saw was a change in the Vmax, but not a change in the Km. And so this told them, right, what they had was a non-competitive inhibitor. And then they did, further things to say it doesn't actually inhibit any of the other polymerases we have in our body. So, it's going to have probably low cytotoxic effects because it's not inhibiting other DNA polymerases. And so does this make sense? One, does it make sense what they did for what I want you to know for the exam? But also two, if you look at this figure they made, does this make sense what they're actually showing?
Aren't they basically showing that the Vmax is decreasing, the y-intercept as a result increasing as they increase the BIRG587 concentration? That's
exactly right. And so if you look at this, right, each one of these lines is increasing as they did a series of reactions, right? This is increasing inhibitor, right? So as you get more inhibitor, then you get more inhibition. So you get the most significant change in Vmax. Okay, so that brings us to transcription. So if we look at gene expression, right, our DNA doesn't directly encode for proteins. We have a step in between. And so we have an RNA step in between. And so for gene expression, right, we go from DNA to RNA, and we call the RNA that's going to encode a protein, we call this a messenger RNA. Right, we have other RNAs that are expressed, they're non-coding RNAs, but if it is expressed, if it is translated into protein, this is messenger RNA. And so how RNA is synthesized from DNA is by RNA polymerase, right? And in this case, instead of using the deoxyribonucleosides, we're going to use ribonucleosides. And again, remember the four we use A, G, C, and U. But just like DNA polymerase, the enzyme goes in the 5' to 3' direction. So it's always going to take the 5' phosphate and put it on the 3' OH. The one thing that's different is RNA polymerases can start adding nucleotides without having an OH already there. So it doesn't have to have a primer to begin. And you can just do the base pairing with an incoming ribonucleoside and then add another one after it and start. And in bacteria, they have one RNA polymerase to do all their jobs. In eukaryotes, we have 3 RNA polymerases. So again, we're subdividing tasks and making it so different polymerases are doing different things. And you should know these 3 for sure. You should know what RNA polymerase 1 is transcribing, right? So the ribosomal genes. RNA polymerase 2 is doing all the protein encoding genes. It also does some few things like microRNAs and some other non-coding, usually larger non-coding RNAs. And then RNA polymerase 3 does the tRNA genes and genes for really small non-coding RNAs. Yeah, and so when we look at the activity, what we're going to see is the enzyme's going to make this short bit of DNA RNA helix. So we're going to have that hybrid helix that's going to get formed by this enzyme because it's going to use DNA as the template to make RNA. But it does a reaction in the end that's just like DNA polymerase, right? It's going to have 2 coordinating magnesiums. It has the aspartates that we talked about coordinating those magnesiums. And one of them is going to facilitate the attack from the 3' OH onto the alpha phosphate. And the other one's going to help stabilize and facilitate the other 2 phosphates for release. And so we talked about how the RNA polymerase knows where to start. So we talked about promoter regions, which are regions of DNA upstream of where it's going to start transcription. And in bacteria, right, we have this region that's 10 bases upstream and 35 bases upstream, right? So the -10, -35 sequence. And so this is where the RNA polymerase in conjunction with the sigma subunit is going to bind. And so this goes along the DNA as a complex. It hits this -10, -35. The sigma binds to that very tightly, holds on. So this is what lets transcription then start there because it has time, it can hold on. It begins, sigma comes off. So once sigma falls off, then it's not being held in place anymore. The polymerase isn't stuck there. So now the polymerase begins to move and synthesize the rest of the gene sequence. Like most things, it's more complicated in eukaryotes. And so we have a lot of what are called general transcription factors. And so these are all required for all gene expression. So for all genes in eukaryotic chromosomes, we're going to require this set of general transcription factors. The things you should know, you should definitely know about the Tata box binding protein. Right, so this is one, it's a subunit of transcription factor 2D. And so it's going to bind to this region of DNA we call the Tata box. And so what it does is it binds as a dimer and it puts a really sharp kink in the DNA. And this gives a structural signal for the other subunits, everything can then assemble there. So it's going to change the conformation in the DNA so that everything else knows this is basically the start place. So Tata box binding protein is an important one for you to know. And so I'll get to another one too, but to turn on, right, or off transcription, we can regulate it through through activators is how we would turn things on, and activators bind at sequences called enhancers. And so these enhancers help the polymerase and everything organize into the right place and be ready to go. We can also have things that help turn it off as well. So we have repressors, and repressors bind the regions we call silencers. And so these can be really far away in eukaryotes. They can be thousands of bases away. And so something has to coordinate everything at that promoter site. And there's this complex called mediator. And mediator takes in these signals from the activators, and it actually physically coordinates it as well. Everything's brought together through mediator. And so lots of activators, lots of repressors all bind to the mediator and coordinate that signal. So we get everything bound at that right place. And so for transcription to start, what has to happen is the C-terminal domain of RNA polymerase. It has this C-terminal domain extension. Transcription factor 2H, it's also a kinase, and it phosphorylates that C-terminal domain of RNA polymerase. And when it does this, this lets the mediator go, essentially. So now we're getting rid of that complex that was holding it there. And then so transcription can start. And there's different phosphorylation patterns that can happen at the C-terminal end. One of those patterns lets mediator go. There's other patterns that then recruit other proteins. And so those other proteins can be RNA processing proteins. So when we talk about the processing of eukaryotic RNAs, it's all happening at the same time with the RNA polymerase. So as the RNA polymerase goes along, those enzymes are already there. And so then the question, we're moving along. So we have transcription elongation. How does it terminate? So we talked about in eukaryotes, it reaches a termination signal and comes off. But in bacteria, there were two mechanisms we talked about. We talked about intrinsic termination. So this is with an RNA hairpin. And then so a hairpin's formed right through this palindromic sequence. And so, right, that's something that can be read forward and backwards. We get this structure from base pairing in the RNA. And then at the end of that hairpin, there's this poly-U sequence. And so the poly-U sequence, what happens is the polymerase, right, is going to have this, going to form that hybrid DNA RNA helix. And so the UA base pairing, so the ribonucleotide U and then the deoxy A together, that base pairing is one of the weakest ones. So once we start to get that formed, those start to come apart. And then so we get that helix, that RNA DNA helix come apart. And then we can get those two dissociate from each other. So then the RNA can come off of the polymerase, right? And that is termination. But we also talked about protein dependent termination. And this is, we call row dependent termination. And so there are these proteins that form this complex, and row acts as a helicase. And so it grabs onto the transcript, it's coming off, and it starts pulling on it, and it works its way up to where we have that helix, that RNA DNA hybrid helix, and it's a helicase, so it separates it, right, and pulls it off of away from the polymerase. So in eukaryotes, we talked about the processing of those transcripts after they're made. And really, like we said, it's kind of happening at the same time as it's being made. But we talked about the 5' cap, the 3' polyadenylation. And then we also talked about splicing. And so getting introns out of those messenger RNAs. But as I said before, all those factors that do that processing all can ride along with the polymerase. So as the transcript is coming off the polymerase, we can already have those happening. So like capping, as we get that 5' end of the transcript made, the enzymes are there and can process and cap the messenger RNA. So once it comes off, it doesn't need to be capped, it's already been done. Right, and the cap is a strange nucleotide. It's 7-methylguanosine. And so we have guanosine with a methyl group added. And then we get this weird 5 to 5' linkage. And so remember, at the end of our transcript, we should have a triphosphate. And so the first thing that happens is one of those is a phosphatase comes along and takes off one of those phosphates. And then we get this guanylyl transferase that makes that 5 to 5' linkage. And then so now we have this built up, and then we have a methyl transferase come in and put on the methyl group. And so I do want you to know the order of these events, right? So removing a phosphate, making the 5' linkage with the G, and then adding the methyl group. And so this is going to provide a place, remember, for the ribosomes to get on to the transcript to begin translating in eukaryotes. So it's going to give us that cap's important to make sure the ribosomes can get there. Also, what will happen is as we finish up transcribing, then we have a poly A polymerase that comes and puts this poly A tail. And so this really is important for stabilization of the messenger RNAs. And this is hundreds of bases along, so a few hundred bases of A's are added. And then another difference between bacteria and eukaryotes is that bacteria genes, right, these are uninterrupted. They're just the DNA is what is going to be transcribed into the messenger RNA, and that's what's going to be used to make the protein. In eukaryotes, our genes are interrupted with sequences we call introns, right, or intervening sequences. And so we have exons, and so this is the bit that's going to encode polypeptide sequence, and interrupted with introns. Then we have other exons, right? They're going to encode the polypeptide sequence, and then interrupted with these introns along the way. And so one thing this does, having all these introns, and we're not going to talk right now about how these are spliced out, which is something you should know, but I won't ask it on the short answer. How these are, when these are spliced out, they don't have to be spliced out. Every intron doesn't have to be spliced out the same way. You can make it so, right, this intron here is spliced out just as it normally here and here, or the end of this intron and the end of this one can be spliced out. And then we actually totally skip this exon. And then we would have this exon and this exon made in the messenger RNA, right? This is called alternative splicing. And so this is where those different patterns of splicing happen in the messenger RNA. And so then actually what we get is lots of different messenger RNAs from the same DNA sequence, because we just have different patterns of splicing out of introns. And so this is why if we look at the number of genes E. coli has, how many different genes E. coli has, that's how many different proteins we see, just about that same number. The same with yeast. Yeast don't have a lot of introns. So if they have 6,000 genes, they have about 6,000 different proteins. They have a few introns, but not a lot. Whereas in humans, most of our genes have introns, and most of those have alternative splicing. So we can go from 20,000 genes in our DNA sequence to say 120,000 different proteins made, because we have different splicing patterns for each one of those genes. And so each one can make multiple proteins. And so we finished up talking about transcription by talking about RNA and the flexibility RNA has, both because it's single-stranded usually, right? And so with being single-stranded, it can base pair with itself. And that single-strandedness just lets it accommodate other base pairings as well. So we get a lot of different, we get base pairing flexibility. And when we form those structures, we can get tertiary structure out of it. Right. And now we're starting to sound like protein folding, right? We have the primary structure, then we get the secondary, and we get the tertiary. We get the same thing with RNA. And so the flexibility and then having that tertiary structure allows RNA to do similar things to proteins, like stabilizing transition states. It can orient things in the right direction. And so RNA can act as enzymes, and it can act as true enzymes where it can catalyze a reaction and then catalyze another one and have turnover in the same way an enzyme does. And the two we talked about as being essential in all organisms are ones that process the tRNA, which is RNase P, and the ribosomes, right? So forming the peptide bond is done by the ribosomes. And so it's a ribosome. Okay, so I'm going to cover one more topic and then end, even if we didn't finish it. So I want to talk to you about the amino acyl tRNA synthetases. Remember what their job is they're going to be like the link between the messenger RNA and the peptide bond that's going to be formed. And so to do that, we have to have some kind of link, and that link remembers the tRNA. And so the amino acyl tRNA synthetase's job is to take a tRNA and hook an amino acid to it. And so we have 20 different amino acids. So they're one, and then we have 20 different tRNAs, so one for each amino acid. And so we also have 20 different amino acyl tRNA synthetases. So each one, its job is to find the right amino acid and hook it to that particular tRNA. So we have an alanine tRNA synthetase, and its job is to take alanine and tRNA ala, or the tRNA that's for alanine, and hook those two together. And remember, it does this as it forms this ester linkage. And it does this reaction, forming that ester linkage, in 2 steps. And so in the first step, it takes the amino acid and ATP, and it makes this, it takes the amino acid and the tRNA, and it makes this amino acyl adenylate. So we have the amino acid with AMP, and then we release the pyrophosphate. And so releasing that pyrophosphate, really almost instantly it gets hydrolyzed. And so that makes this reaction, mostly irreversible. And then the next step, it takes that amino acyl adenylate and the tRNA and hooks those two together, and then releasing the AMP. And so in this reaction, right, we required, right, ATP got hydrolyzed and pyrophosphate released. So the energy from the ATP now is used to form this ester bond. And so you should definitely know the overall reaction in the 2 steps. And can you, right, shown on this slide, right, is both, right, just the equation of this, but then also written out. So could you write it out? Could you explain it? Either way, there's things you could do as a short answer question. Okay, so we talked about the different classes of synthetases and how class 2 attaches to the 2' OH, or sorry, class 1 attaches to the 2' OH and class 2 attaches to the 3' OH. And it actually doesn't matter that this happens this way and that they're different, because we get transesterification. So this amino acid can actually freely move between these 2' and 3' positions. And so in the cell, we actually see that. You can look at the any particular amino acid, and you could find a population of tRNAs with it at the 2' and a population with it at the 3'. Okay, so the synthetase's job, remember, is to pick the right tRNA and the right amino acid and hook them together. So that means it has to pick the right substrates, right? It has those 2 substrates it has to pick. And so the tRNA, what it recognizes usually is the anticodon and the acceptor stem. And that's because the anticodon, right, this is where we have base pairing with the codons, and the codons define what amino acid will get put in. So this is an obvious choice because that's telling you, what amino acid's there. So often synthetases recognize the anticodon loop. The ones where it doesn't are usually the ones where there's like 6 codons that could code for the same amino acid, because then it starts to get to be too structurally different for it to recognize that. But all the other ones, for the most part, they do. But all the synthetases recognize this acceptor stem. And so it's going to recognize the acceptor stem and know that it's the right tRNA. For the amino acid, it's a lot harder, but it still has to recognize the right one. And we talked about phenylalanine and tyrosine, where the difference is, right, that one hydroxyl, and it has to distinguish between them in the different ways it can. And we also talked about the 3-anenyl tRNA synthetase, which uses a coordinating zinc ion. And so the OH of the 3-enene is going to form a bond with that zinc ion. And so this is one way to distinguish it from other amino acids. So something that's really structurally similar, like valine, could be distinguished. But something like serine maybe can't be distinguished so well, because it's going to be able to form this hydroxyl on the serine can form a bond with the zinc ion. And so about half of the synthetases have what's called an editing function. And so they actually have 2 active sites. They have an active, right, or activation site. And then they have another active site, which is called the editing site. And so what happens is the CCA end of the tRNA with the amino acid, if it's the wrong amino acid, it can swing into the editing site, and this amino acid can be hydrolyzed off. And we think about this as a double sieve mechanism, where the first sieve is the activation site and the second sieve is the editing site. And so remember, a sieve's like, think about it like netting, 2 different sides of netting, that one after the other. And so the first sieve, what happens is at the active site, any amino acid that's bigger than the correct amino acid is excluded. Right? So anything larger can't get into the active site. And so it's been essentially sieved out. And then anything that's smaller, so the right amino acid or anything that's smaller, can get in the active site and get activated. But then we have an editing site, which is like another layer of that sieve or that netting that's a little bit smaller. And then in this case, your amino acid that you want is now too big to fit in the editing site. And so anything that's smaller gets put in the editing site and then can be hydrolyzed off. And then so if you hydrolyze those off, now what you're left with only is your correct amino acid. Does the double sieve mechanism make sense? Or would you like me to go over it again?
Yes, I think it makes sense, especially with the trash can analogy.
Okay.
So do we just need to know like how it has pretty much 2, I guess editing site has an active site and an editing site, and then what they do, or do we need to know specifically about the CCA arm?
You would need to know that the CCA arm with the amino acid moves into one site and then into the other site. yeah, the swinging of the CCA arm is important in that mechanism.
Okay.
Because
Thank you.
the amino acid is on that, right? It's attached to there. So that arm has to move from one to the other. So describing the double sieve mechanism, right? Something like that for a short answer question. Yeah, and how that can lead to, right, higher fidelity, because you're getting rid of either the big things or and then the smaller things. Okay, so I need to go. It's almost 3 o'clock. There's a few more topics, right, covered on that study guide, but I think we got through a lot. Hopefully it helps. Are there any questions?
So we were talking about RNA polymerase or DNA polymerases. Which one was there a DNA polymerase that acted as a primase for bacteria? I don't see it on the.
So no. Wait, let me get back to it. So some of those that are DNA polymerases that then act as a primase, right? They're actually a complex, and they're going to technically have one of their subunits that works as an RNA polymerase, because remember, primase is it's going to make a short stretch of RNA.
Okay, so there is no primase for bacteria though?
No, there is one.
And what would that be then?
So in that case, I just call it primase. It wouldn't be included here because it's not in a complex with a DNA polymerase.
Okay.
Does that make sense? Because here we're just listing the DNA polymerases, and DNA polymerase alpha also has an RNA polymerase activity that's used as primase. Whereas in E. coli, it doesn't have a DNA polymerase that's has another activity as an RNA. And so it's not called DNA polymerase. So yeah, this is, I guess, just for you, because like DNA polymerase alpha it's a heterotetramer. And so like 2 of the subunits are actually a DNA polymerase and 2 are actually the RNA polymerase primase subunits. So both are there in that complex.
Okay, thank you. And then if no one else has any questions, could we also review the 7 main features of B form of DNA that you want us to know for the exam?
Yeah, right, so these are your
We're still on, right there. Okay. There you go. So with double helical here, this is for B form, right?
This is for B form. And I think the next slide, maybe, I can't remember. I think maybe in the I can't remember where I went over it in the lecture, where I also showed the Z and the A form. But this slide here, this double helical structure of DNA, this is for the B form.
Okay, awesome. Thank you.
You're welcome.
Would this be more of like a list of 7 main forms, or would it be more of like mix and match with like multiple choice type questions?
I haven't decided. I kind of like it as a list, but like it's really just knowing the features of the double helix. And so I think this is probably a list type thing. But then if it is a list, then you're not going to be writing about each of the things. You're not going to write, right, they stack on top of each other and the van der Waals help stabilize and the edges are exposed, right? You would just say that we have base pair stacking perpendicular to the helix.
And then one last question, if that's okay, Dr. Reynolds.
Yeah.
Sorry for going over, but when it comes to terminating or translational termination differences between bacteria and eukaryotes, I guess what's the expectation in regards to detail there? Like, is it enough to just
So,
say, yeah, go ahead, sorry.
sorry, do you mean, so on the study guide, I have, right, we have bacterial transcription termination. So knowing the difference between intrinsic and row dependent. And then for translation, I want you to know initiation.
Okay, so we need to know the differences in initiation.
Initiation, right. And so
Okay.
remember, I'm trying to get to the slide. Remember we have like a special initiator tRNA, right? Methionine is used in both bacteria and in eukaryotes, but bacteria has that special form of methionine. Hold on.
And the special form of methionine is on that tRNA, correct?
Yep, and it's oh wait, here we go, sorry. So actually it gets, right, that's exactly right. So it gets the tRNA, the formal methional tRNA gets a methionine put on it. Once it's on the tRNA, then we have it turned into formal methionine. And
Okay,
remember
so
what bacteria have, right, is a sequence, yeah, Scheindel-Garner sequence, where what we get, right, is we get the small subunit, the initiator tRNA with formal methionine. We get that brought there by an initiation factor 2. It's going to bind to that Scheindel-Garner sequence, right? The ribosome is now bound. And when it's bound, then we have that initiator tRNA bound into the P site. And then when the large subunit assembles, it's all ready to go, right? We're at the start codon. And now we're ready to bring in a new tRNA to the A site. So now we've set up for what the reading frame is, right? That initiator tRNA bound to that correct start codon because the Scheindel-Garner sequence put the ribosome in the right place.
Okay, so the Scheindel-Garner sequence puts the ribosome in the right place in bacteria. And also we have the formulation of methionine. Okay.
Yep. And that's what that formal methionine does. It makes it so it can't be used anywhere else, because that formal group won't let it be added to the C terminus of another amino acid. So it has to, and that has to be start. Right? That has to be the first. It can't be included anywhere else in the protein. And then
Okay.
remember in eukaryotes, right? With eukaryotes, we have a different thing. We get this complex formed separately from the messenger RNA. We have the small subunit, an initiation factor, an initiator tRNA bound with methionine or charged with methionine. And this binds to the 5' cap. And then it moves along until it finds the first AUG codon. And when it finds the first AUG codon, it pauses and that initiator tRNA is in the P site. Now we can get dissociation of initiation factors and the large subunit binding. And then again, we're set up for the right reading frame. We have an initiator tRNA in the P site bound to the start codon. And so now we defined, right, this is the reading frame. And actually, that's where I wanted to get to. So that was the last slide I have.
I had a question about the specific initiation factors. Do you want us to know like specifically that like EIF4E does this, or can we just know that these processes are being carried out by initiation factors?
Sort of both.
If that makes sense. Okay.
Yeah, I want you to know initiation factor 2 in E. coli. That's because when it brings it there, it's gonna right? Hydrolyze that GTP. So that's good. I want you to know EIF2 in eukaryotes.
Okay, thank you. Is that the same for like the elongation factors too, and like for termination?
Yeah, I want you to know EFTU, and I want you to know EFG.
Okay, thank you.
And then so technically, right, if we think about that, then we could start to talk about how many ATP and GTP are used to make a peptide. Right, so let's think about it. If, let's just say we're going to make a peptide that's like 10 amino acids long. Right, so just for translation, let's think about how many ATP we need. So we would need one for each of the tRNAs to get charged, right? So we would, at a minimum, we need 10 ATPs to be hydrolyzed to get the charging of those 10 amino acids on the tRNA. We would need a GTP to be hydrolyzed by initiation factor 2. I think I have a GTP. Then remember, EFTU is going to hydrolyze GTP for each cycle. Let me see if I have that. Maybe this is better, right? So remember, EFTU brings the tRNA with GTP to the A site. It's going to hydrolyze GTP. So each one of those tRNAs that get brought, now we get a GTP hydrolyzed. And then we talked about, right, EFG and it hydrolyzing GTP to terminate, right? And so you get this cycle, one GTP for that initiation factor, one for the termination, the EFG, and then one each for the EFT use. And so you could think if it's 10 amino acids, we would need at least, or let's just say about 12 GTP hydrolyzed, right? And then for ATP, you'd have about 10. Does that kind of make sense?
Yeah, thank you. Where does it
I think
use...
sorry, go on.
Oh, no, sorry. I was like, where does it use ATP instead of GTP in that? Because I thought we would all used.
Well, just on the ribosome, it wouldn't, but the synthetases each use an ATP to charge the tRNA.
Okay, gotcha. Yeah. Okay,
That's
thank
it.
you.
So what I want you to see here, I will for sure ask probably true false, but some question about EFTU hydrolyzing GTP and how it has to happen, you know, in each cycle and how many GTP would be used. I may ask something, not specifics. Let me see if I can find it. I probably will ask some sort of multiple choice question about how EFTU and the ribosome are going to make sure we have the proper base pairing. I'm not going to ask you specifics about like A1492, but understand that the ribosome's using base pairing, right, with the or looking at the base pairing between the tRNA and the mRNA. And when it's proper, we get a conformation that allows GTP to be hydrolyzed by EFTU. And when it's not a proper base pairing between the codon and anticodon, then what happens is we don't get a structure change that allows GTP to EFTU to hydrolyze GTP. Does that make sense?
Yeah, that helps. Thank you.
But that would be some sort of like multiple choice or some kind of question. But I don't want you to overlook that. I want you to understand that it's monitoring those interactions and then releasing the tRNA when they're correct. The other thing about this was that, remember, it looks at what it's monitoring really is the first two positions. And the last position, remember that codon we call the wobble position. And that's why in the third position, if we look at the codon table, a lot of times the code for the same amino acid, those codons vary in their third position, but not in the first two. Okay, so I need to go. If you have questions, you can email me. Let me get out of here.
Thank you, Dr. Reynolds.
Yeah, thank you. Yeah, let me know. And I hope you have a good weekend. And I will get this,
Thank you.
I'll try to get this posted reasonably soon. All right, thanks.